All right, part 2, understanding the stages of mitosis and meiosis. So one thing to remember is that there's 2 different phases. There's mitosis and meiosis, and why are there 2 phases? So they both start at the same point. Interphase has completed, DNA has replicated, cell has grown large enough to divide, and so now we need to divide DNA into the appropriate daughter cells. And so there's 2 goals. One goal is to make 2 cells with 2 exact DNA copies of each other. So in development, as a result, you want to have 2 exact copies. So skin cells are identical to each other, heart cells are identical to each other. You don't want a heart cell to have a different complement of DNA. So the goal of mitosis is conservative. It's to create 2 cells with the exact same copy. Meiosis, the goal is to not create the same cells with the same DNA, but it's rather to create new cells with different amounts of DNA. And further, the goal is to half the amount of DNA, to go from a diploid cell to a haploid cell. And so this is used in the germline. So there's 2 steps that are necessary in meiosis. One is to become a haploid cell, and so this is so that a sperm cell and an oocyte can join together to create a diploid cell. So becoming haploid is one of the goals. And then you also want each sperm cell or each egg cell to be different. So if you have siblings, you know that your brother and your sisters are very different from you compared to identical twins. And that's actually a goal, an evolutionary goal, is that you actually want to create a random mixture of DNA in all of your offspring. So you don't want all your offspring to be exactly the same. And so meiosis accomplishes both of those goals. It randomizes DNA and it also creates haploid cells. So your textbook lists 5 stages of mitosis: prophase, prometaphase, metaphase, anaphase, and telophase. Typically, most textbooks do not include the prometaphase. Typically, it's just prophase, metaphase, anaphase, and telophase. So for the exams, I only expect you to know these 4 phases. So prophase is preparation phase. Prometaphase, we won't talk about. Metaphase is the midplane phase. Anaphase is the active phase, and telophase is the termination phase. So it's often easiest to understand what is going on during these different phases by actually staining DNA and staining microtubules. So an interphase, as we discussed before, interphase is before mitosis or meiosis has begun. The chromosomes do not exist, so the DNA is in an extended and uncoiled fashion. So if you stain DNA, you would just kind of see these blue lines throughout the nucleus. And then you would see kind of microtubules throughout the rest of the cell. So the nucleus is kind of right here. So you can see all the DNA within there, but you don't see any sort of structure that looks like a chromosome. So prophase is the phase when the chromosomes actually coil up and condense. So you start to see, instead of just being distributed blue throughout the nucleus, you actually start to see structures that are going to eventually look exactly like chromosomes. So this is when the proteins are aggregating the DNA, they're condensing the DNA into a much more condensed state. And the goal of prophase is kind of to prepare the DNA. The P is for prepare, is to prepare the DNA in order to be able to be segregated. So prometaphase, the chromosomes are clearly double structures. Centrioles reach the opposite poles, and then the spindle fibers actually form. So the centrioles are the organizing location for the microtubules, and so this is what determines where the chromosomes will be separated to. So some of the chromosomes will be split and then sent to this centriole, and then the others will be sent to this centriole. So these are organizing structures for microtubules. So you see these microtubules kind of form from there. And so if you looked in the cell, you would start to see that. You would start to see these red lines kind of like go toward these center points. So you can kind of group these 2 phases together, prophase and prometaphase. I won't ask you to actually distinguish between these. So metaphase is the point where the centromeres actually align on the metaphase plate. So the M for metaphase I think of as the midpoint of the cell, so the midplane of the cell. So you can see all of these chromosomes have not only formed, but they're now actually moving to the midplane of the cell. And so this is important for actually segregating all of the different things all the different DNA to the different size. Anaphase. Anaphase is the active phase. So this is when DNA is actually being moved from one side, from the midplane to one cell. So this is when segregation is actually occurring. And so the chromosomes, the DNA is actually moving along these microtubules towards the centrioles. And so the way it works is that, the spindle fibers has connected to the kinetochore, which is at the centromere region. And so this is when the chromosomes actually split. And so you actually degrade the cohesin, the suggestion. And so when you lose the cohesin connection between the sister chromatids, you can actually now segregate and split the DNA molecules into different cells. And so this is part why mitosis is conservative, is because we talked about before, each of these sister chromatids are identical DNA molecules to each other. And so because they occurred from DNA replication, you now get one exact copy of each. So, one copy produced from DNA replication goes to daughter cell 1, and the other one goes to daughter cell 2. And so the active phase is when the actual splitting occurs. And then finally, telophase is when the termination occurs. So the nucleus reforms. So you now have a nucleus to hold the two new DNA contents of the cell, and cytokinesis commences. So now you're actually going to split the cell into two different cells. So meiosis is different from mitosis. And so there's a couple main factors that distinguish why they're different. So again, 2 rounds of meiosis are necessary. So this is the first time actually we covered this. So the result of meiosis is you have 2 diploid cells. So each of these cells each contain a mother's copy of chromosome 1 and a father's copy of chromosome 2, and so on for the rest of the chromosomes. So the result is diploid cells. And so in order to actually create haploid cells in meiosis, you actually need to undergo 2 rounds of division. So you have a first division, which is called Meiosis I, and that results in these 2 cells. And then you have a second round of division that results in 4 haploid cells. So the result of mitosis is 2 diploid cells. The result of meiosis is 4 haploid cells. And again, that's because there's 2 rounds of division. So that was one of the goals, is to create haploid cells. And then the other goal is to actually create genetic variation. And so how does Meiosis I create genetic variation? So one of the important parts of creating genetic variation actually occurs in the first phase of Meiosis I, so in Prophase I. And so the other thing that occurs is that actually homologous chromosomes find each other and join together and undergo DNA crossover. So we'll see that in a little bit more detail. So this occurs during Prophase I. So Prophase I is the first prophase stage during Meiosis I. And so not only do the chromosomes condense, not only do the chromosomes form due to condensation of the DNA, but actually also what happens is the homologous chromosomes are able to find each other. And so you have your father's version of chromosome 1, you have your mother's version of chromosome 1. And so in mitosis, these are in just completely different parts. So if we looked in mitosis, they would go to the midplane and they would line up, completely separate from each other. So they'd be completely independent from each other. But in meiosis, these actually find each other and actually come together. And so this forms what's called the tetrad. So the tetrad occurs when you have 4 chromatids, 1, 2, 3, 4, because you have the 2 homologous chromosomes joined together. And so not only do you have these things form together, the homologous chromosomes come together to form the tetrad, but you actually have DNA exchange that occurs. And so parts of your father's chromosome 1 go and become fused to your mother's chromosome 1 and vice versa. So you can see this is occurring. There's initially a crossover that occurs where they kind of come together, and then eventually you actually create a new chimeric chromosome that is parts of each. And so this is part of the process to ensure that each haploid cell has a different complement of DNA. You're rejoining DNA in unique ways in all of the different meiosis, in all the different times that meiosis occurs. So because prophase I is such a complicated stage in meiosis, it's actually further divided into 5 different substages. Leptonema, zygonema, panchenema, diplonema, and diakinesis. And so initially you have the chromosomes coming together, the bivalent forms during zygonema, the tetrad forms during pachenema, the chiasma form, so this is when you actually have crossover occur where one of the homologous chromosomes invades the other, and then finally once you have that resolves, and then there's breakage, there's double-stranded breakage, the DNA molecules are fused together, and then that occurs during diakinesis. So after that, meiosis I results in the creation of 2 cells. And so these actually, instead of what's important to note during meiosis I, so the metaphase, anaphase, and telophase are kind of very similar. So in metaphase, the tetrad goes to the midplane. Anaphase, you have separation. Telophase, you have termination, where the nucleus reforms and you have 2 cells form. But what's important to note in meiosis I, what actually separates is not the sister chromatids. So remember, mitosis I, the sister chromatids separated. Now what we see are the homologous chromosomes separating. So one homologous chromosome from each cell, from one homologous chromosome goes to each of the cells. And so you'll notice that these homologous chromosomes have been modified due to the acts of recombination. So you have a mostly blue one, but now you have parts of orange because the father's chromosome has been incorporated into the mother's chromosome and vice versa. And so these are the results of prophase II, so of telophase of meiosis I, and you can already see that these cells have different DNA contents. So this cell right here has a lot more of the father's chromosome 1, and this cell right here has a lot more of the mother's chromosome 1. So it's already not conservative, and you've already created variation between the DNA content within the cells. And then meiosis II is the goal of meiosis II is then just to kind of follow up to create haploid cells, and the way it does that is it splits the sister chromatids apart. So in metaphase, now you've aligned all of the chromosomes at the midplane, and in anaphase, you've actually split these apart, split the sister chromatids apart so that each move 1 to each cell. And then telophase occurs, and now you've created haploid gametes as a result of that. So why meiosis but not mitosis generates variation? So there's 2 main reasons. So one is recombination. So this creates variation within a chromosome. And so we saw this before, where now we have new versions of chromosome 1 where they're fused between the father and the mother's complement. And so if we looked at a completely different meiosis, where that recombination occurs is completely random. And so you have completely new chimeric structures being formed every time you do a meiosis. The other reason is that homologous chromosomes are randomly segregated at anaphase 1. And so when these homologous chromosomes are separated, in this case, chromosome 1 that is mostly from the mother is paired with a chromosome 2 that's mostly from the father. But in a different meiosis, you could have a completely different situation where this one and this one, are paired together into the cell. And so because of that, all of the products of meiosis, are very different from each other. What about the sex chromosomes? So you might think, the XX makes a lot of sense during meiosis, but what about the XY? What happens to the XY? These are very different sizes. So the X chromosome is much larger than the Y chromosome. They have very different genes on them, but they actually come together and form their own pair during meiosis. So the tetrad is formed so in male individuals, a tetrad is formed between the X chromosome and the Y chromosome. And that's because there's actually these pseudo-autosomal regions that allow pairing to occur. And so there are very similar DNA sequences in the X and the Y chromosome that allows these to come together to form the tetrad. And so there is actually recombination that occurs within these pseudo-autosomal regions. But in the most case, this has very little effect on the rest of the chromosome. So as a result, recombination isn't very important for the X and the Y chromosome, so you don't create these weird fusion chromosomes that are part X and part Y. And so the homologous chromosomes separate during meiosis I, so the X's go to one cell, the Y's go to the other cell, and then the sister chromatids separate. And so you have 4 gametes, one with an X chromosome, one with an X chromosome, one with a Y, and then one with a Y. So the formation of gametes in mammals. So these are actually the precursor cells, the spermatogonium and the oogonium. I'm sorry, I'm not a developmental biologist. I could be butchering these names. But these are just the cells that are actually forming the sperm and the ovum. And so in male cells, so once they they've undergone DNA replication, and so then they need to divide into 4 haploid cells. So the primary spermatocyte is what, this is prophase I. This is an example of what the cell looks like in prophase I. And the tetrad has form, and then as a result of meiosis I, you create 2 secondary spermatocytes, and these spermatocytes undergo meiosis II to form spermatids. So these are 4 haploid cells, and then these haploid cells differentiate into the sperm cell. So they have to grow the tail, they have to do other things that are necessary for a sperm cell to be a sperm cell. It's a little bit different in females. So there's the primary oocyte. The primary oocyte is an important term just to know for developmental purposes. And it is in prophase I, so the tetrad has formed. And then meiosis I occurs, where you split the homologous chromosomes, and there's actually a secondary oocyte formed and a polar body that is formed. So in female development, this polar body doesn't actually undergo meiosis II. It actually just gets killed and lost. And then the secondary oocyte forms an ootid and a second polar body, and then the second polar body is degraded, apoptosed, and dies. So only one of the haploid cells is actually able to undergo differentiation into an ovum. So it is thought that this is actually a quality control mechanism. So it helps ensure that the oocyte, the ovum that is formed from a healthy DNA structure. So just to summarize the differences in mitosis and meiosis, so mitosis in somatic cells, and the goal of it is conservative, to have the exact same copies of DNA. If we look at the DNA for nucleus, we have the S phase occurs, synthesis occurs, and then eventually mitosis completes, and then you have 2 cells with 2 DNA per nucleus. Whereas the goal of meiosis is to form 4 haploid cells, and so you have duplication, and then you have meiosis I, and then meiosis II, so that you have the content of DNA in each cell. And then an important part of meiosis is the tetrad, recombination between the tetrad, and then that promotes variation within the DNA content of the cells.